After listening to the heavy talk on treatment or the failure of it in majority of cases, I'm going to provide some different side to the story that we might be starting too late, doing too little, and I think we missed the boat. So I'm going to talk about a theory of dual teratogenesis. Clearly it relates to intrauterine programming, two pathways which can lead to diabetes. And as I say always, without tiring out, mine is the only diabetes unit anywhere in the world which has mother and baby as the logo. And that's because of the firm belief we had for last 30 years that improving the health of the young girls will improve the health of the next generation. There's also the second logo is the modern Ganesh. And as I say, when I was a child, this Ganesh used to travel on the back of this mouse. And there was a disparity in the size of the vehicle and the passenger, so travel was very slow. Today's modern Indian Ganesh clicks the mice, and he moves very fast, and that's the rapid transition. So a combination of these two factors, the epigenetic programming and the rapid transition, have brought on the epidemic which we are facing. So this is the dogma which we all learn. I learned that the susceptibility to diabetes is polygenic and therefore non-modifiable. And we usually deal with precipitating factors which are predominantly related to age and obesity. The average participant in the diabetes trials in the world, or prevention trials, is middle-aged, obese, and has impaired glucose tolerance. We use changing cut points to define them one or the other, but they are all end stage for three conditions: for aging, for body weight, and for glucose intolerance. And we treat them and call it prevention, and I think this is the single most problem which has really taken us away from the real prevention. So it's end stage, and very importantly, mostly it's post-reproductive. Being post-reproductive, whatever benefits we see are not passed on to the next generation. The usual advice is to eat less and walk more. And at the end of the talk, I might convince you that the population which we study, if the village women had eaten less and walked more, there would be more diabetes in India. So is there a non-genetic susceptibility factor to diabetes? And that's what has driven our research for the last 30 years. And the ideas came from classic studies. I just now spoke in the other hall about the Dutch winter hunger, where exposure to intrauterine hunger during the Dutch winter hunger either increased or decreased risk of obesity depending on when you were exposed. If you were exposed in third trimester, the obesity risk is less. If exposed in first two trimesters, the obesity risk is more, because the number of fat cells is determined here and brain programming happens here. Same time, data from Pima Indians showed that if you were exposed to diabetes in utero, then your risk of obesity and diabetes is higher later on, compared to the babies which were exposed to mothers who developed diabetes later or never developed it. So it was not genetic, but it was intrauterine exposure. This led to various theories, and diabetes begets diabetes based on the Pedersen-Frankel idea that excess transfer of nutrition to the baby will stimulate beta cells into hypertrophy, secrete more insulin, which in short term will produce a bigger baby, neonatal hypoglycemia, but long-term obesity and impaired glucose tolerance. And very provocatively, he called it teratogenesis for the long-term effects. So within 10 years, we had idea that undernutrition in utero as well as overnutrition in utero can cause diabetes later in the life. And this was rationalized when David Barker published this paper. He showed in 91 that birth weight is a predictor for your risk of diabetes. If you are born lower birth weight, the risk of diabetes is higher. It's nothing to do with low birth weight as defined by WHO, 2.5 kg. Lower the birth weight, higher the risk. So that if you were born more than 9.5 pounds in England in 1930s, your risk was almost 5 times less than if you were born less than 5.5 pounds. He called it a thrifty phenotype hypothesis. And I can say this publicly, I suggested that name to David Barker when he first met me in 1991, that you should call your hypothesis thrifty phenotype. What he said was that type 2 diabetes is the outcome of the fetus and early infant having to be nutritionally thrifty. That was not enough. Soon a study was done in monozygotic twins. Now monozygotic twins have identical genotype, but if they have different birth weights, the smaller of the monozygotic twin has a higher risk of diabetes. Again indicating that over and above genetics, we have an additional factor of intrauterine programming driven by nutrition. And this led to the concept of either programming or teratogenesis, which is a developmental adversity restricting plasticity. These are all developmental biology terms. What it means is that if you have plenty of everything, you will develop well. If there is an adversity, there is a restriction. And that is called programming or teratogenesis. And it remains with you lifelong because it affects structure or function. And the windows which are important are pre and periconceptional. That's a very important window. Then pregnancy itself, then again lactation, adolescence, wherever development happens, that time, if you influence the structure or function, it remains with you long time. Various causes, nutrition, metabolism, stress, pollutants, and this might be reversible. And that's why it's really important to study and see whether it can be implicated. It manifests at organism level, like what we study, diabetes, etc., but can be traced back to molecular changes. And 3 mechanisms which are currently studied, I'm sure more will be described, will be DNA methylation, histone modifications, and miRNAs. And all these studies are now happening in the cohorts which we have developed. So now we can say that there might be an additional epigenetic modifiable susceptibility to diabetes, which might change the picture, rather than trying to treat the adults and call it primary prevention. And this programming is peri-reproductive, therefore we need to treat as early as possible. And these 2 pictures are in our Pune cohort 30 years ago, and this lady's 2 daughters here are moving on the mobile sort of scooter. And that's the jump which I'll show you what it has done to them. So this actually is also a very interesting worldmapper.org, a site which produces what are called cartograms. And cartogram is a world map where size of the country is proportional to the burden of that condition. So India is big in diabetes, but it's even bigger for early life undernutrition, measured either as low birth weight or under 5 undernutrition. India produces largest number of low birth weight babies in the world, and Indian babies are the smallest at an average birth weight of 2.7 to 2.8 kg, and whole lot of nutritional factors are described here. Whole lot of macronutrients are studied in this condition. This is very interesting, and I've called it history, legacy, and diabetes. This is the map of diabetes increase in last 25 years, which was published last year in Lancet. There are 5 papers in 1 issue of Lancet from Public Foundation of India. You should all read them. This is the diabetes paper. And red indicates the states where the increase in the age-adjusted prevalence was highest. And you can see These are the states which face some form of adversity either just now or in recent past. Be it political instability, the geographic terrain, etc., poverty and socio-economic deprivation. These are the states which have had highest increase in the prevalence. If you look at Mohan's INDIAP paper, in 8 of the states, the prevalence of diabetes in the cities was higher in the lower socioeconomic than in the higher socioeconomic. And this is a very interesting website about height. It's an NCD collaboration on height. They have traced height of different populations over last 200 years. I have shown 3 European countries and 3 Southeast Asian countries. Europeans gained 15 centimeters of height in last 150 years between 1830 to 1980. India remained flat. Some of the Southeast Asian countries even lost height. If you open the diabetes atlas of the IDF, they will call Southeast Asia as the epicenter of diabetes. It's not the obesity capital, it's the stunting capital of the world, at least for the last 150, 200 years. And it should actually wake us up that our history and legacy influences our diabetes risk, and we have completely ignored this. A Danish philosopher said this. He said, life can only be understood backwards, but it must be lived forwards. And Steve Jobs said, you cannot connect the dots looking forward, you can connect them looking backwards. So they already realized that things can be understood only if you look backwards. We set up this Pune maternal nutrition study in 1993 after my initial interaction with David Barker, and one study to show that low birth weight mattered in India. So we enrolled more than 2,500 women in our cohort, followed them every month. 814 of them delivered during our study. They were studied intensively in pregnancy. Their husbands were studied and baby's growth was monitored. At birth, we had detailed measurement of anthropometry. We measured growth every 6 months till 18 years of age, and then we measure it every year. And every 6 years, we do a detailed analysis or a study in both the children and parents, which covers their size, body composition, nutrition, cardiometabolic risk, cognition, etc. We have one of the best follow-up rates in the world, 90% or 95% and above, even after 20 years of follow-up. And we have a blood bank which I developed right from the beginning. So we have extensive documentation of the biological specimens which we can go to and which we do go to for various tests. The average mother in the villages was 42 kg before pregnancy and had a body mass index of 18.1. Her baby was 2.7 kg, and if we use international criteria, 70% of the babies were SGA, small for gestational age, growth retarded. This is a predominantly vegetarian population. Intakes were about 75% of recommended allowance, which was about 1800. Carbohydrates form large part of the energy. Lower intake of leafy vegetables, milk and fruit, and they had a substantial physical activity during pregnancy. Like you can see here, this woman carrying these 2 pails of water. Each one is 15 liters. The village is 1.5 kilometers away. She is only 47 kg at 7 weeks of gestation. And there was no increase in intake or reduction in physical activity during the gestation. Gestational diabetes was less than 1%. It was 0.3% measured by the WHO criteria. And the babies were 2.7 kg. Based on our previous work, we knew that if we compare them with European populations, we get more. So we compared that baby with babies born in UK. You know all this story, I have said this again and again, that they were small in all respects, but were nearly as normal for subscapular skin fold. So they were thin in all other measurements, but they were fat, and the cord blood concentrations of leptin were high, insulin was high, and adiponectin was low. The classic risk factors which we use in adults to predict future risk were already present in the cord blood. Editors of journal are not very fond of simple tests, so they said anthropometry, what does it, can you really measure it? So we did MRI in 30 babies, compared them with full body MRI in English babies. And all three compartments of abdominal fat were higher in the Indian babies, even though they were 800 grams lighter. So that's the thin fat Indian baby with high levels of risk factors in the body composition as well as in the cord blood at birth, so clearly developed during intrauterine life. And a whole lot of maternal influence on baby. Even though glucose tolerance was normal, glucose was still a significant predictor of different measurements in the baby, as were cholesterol and triglyceride concentrations. And certain micronutrient-rich foods like green leafy vegetables or red cell folate concentration predicted better growth, while high homocysteine predicted poor growth. And this high homocysteine came predominantly from B12 deficiency rather than folate deficiency. So we had this peculiar picture of low B12 and high folate. And when these children were followed up, we could relate low B12 and high folate in the mother with increased insulin resistance, increased adiposity, and with some of the neurocognitive outcomes. Now I have Dr. Bere with me, who is a Wellcome Fellow, and he uses high resolution MRI and functional MRI to study brain of these children. And we have now found very interesting findings of differences in growth of different parts of brain and connectivities on functional MRI in relation to intrauterine B12 exposure. So that's a big thing coming now. All this looks like it's partly related to vegetarianism, which in India is multi-generational, unlike Western world. Started with Samrat Ashok, who became Buddha's disciple after the Kalinga War. Religions support it, and very interestingly in Pune we found better education, higher income and higher hygiene predict B12 deficiency. So urban middle class have twice as B12 deficiency as urban slums. Can you tell me why? Pardon? Okay, so the only source of B12 in nature is microbes. And we are all trained to keep the microbes away. And down with microbes is a usual philosophy. So I think that might be contributing to this. We are studying this. Folate is good because it's vegetarian product. So if you have higher food intake, better education, higher socioeconomic status, folate is good. So that creates an imbalance. National programs provide iron and folic acid, and obstetricians give folic acid to prevent neural tube defect. We are not going to criticize them just now, but there is a problem there. And it affects one-carbon metabolism, which is a complex metabolism transferring methyl groups in different reactions. There are 80 methylases in the cell, 80 enzymes which require methyl group. So it's a very central mechanism, and it affects DNA synthesis, repair, methylation, which is epigenetic mechanism, genomic stability, protein synthesis, and other physiological processes. So this is a very central metabolism to learn. And we have published extensively about microbial characteristics of Indians, their association with diabetes, and effects of B12 supplementation on methylation, etc., etc. I'm not going to get into details of this. When these children became 18-year-old, half of them were underweight. That means they had a body mass index less than 18.5. Only 5% were overweight or obese. But 28% of them had pre-diabetes by IDEC criteria. So at an average BMI of 19.1, 28% were pre-diabetic. More in boys than in girls. And we are able to trace them back because we have been studying them every 6 months, so that we can now show that those who are pre-diabetic at 18 years of age had lower disposition index at 12 years and at 6 years of age. So their beta cells were not behaving fully right from early age. And we diagnosed them at this time. Therefore, we try to do a whole lot of studies. But because we can look backwards, we are able to say it was all present even in childhood. And the combinations which produce problem is if you are born small but became big, you are more insulin resistant. On the other hand, if you were born big but remained thin, your beta cells worked better. And if you are born small and became big, you had high glucose. So in other words, if there is a mismatch in the growth between intrauterine life and later life, that's trouble. A baby which is programmed as undernourished, if it is overfed in later life, then there is a trouble. And pediatricians attacked me when I published these results, but now there is a uniform agreement that this is true, and therefore they have tapered their enthusiasm to overfeed the small babies. We can do something else which no one else in the world can do. We can generate first intergenerational glucose curve in the world. So this is F0 mother's pregnancy, which is what we studied in 1993, glucose tolerance test. Then child was born, this is about girls, 6 years, 12 years, 18 years, and pregnancy. So this is 150 first pregnancies in the F1 generation. What you can see is there is a strong tracking of glucose from pregnancy back to 18 years, back to 12 years, back to 6 years. So what we call gestational glycemia in pregnancy is already programmed before. And these girls are moving higher. So I have now challenged the concept of gestational diabetes. That is already there. And just now you listened to me in the other room. Therefore, these girls' ova are exposed to abnormal metabolism right from their early age. We wake up in the 3rd trimester, fight about the criteria for glucose tolerance, call them gestational diabetic, and zap them. Literally, we starve them because we want to reduce the size of the baby. Indian baby is on average 2.7 kg. It's all right in Northern America, where average baby in a diabetic pregnancy might be more than 3.5 kg. Here, we are, I think, required to now revisit the idea of gestational diabetes, around which a very big industry has developed. So I think I become very unpopular by saying this, but I have said gestational diabetes is not gestational, starts very early in life. And whether it's diabetes, we don't know, because every day we change the criteria. And we diagnose it in 3rd trimester when the horse has bolted. So don't get away by actually thinking that you are doing great service if you are diagnosing gestational diabetes in 3rd trimester and producing a small baby. I'll show you when we follow these babies what they are. So this, I think, is important. Another thing is there is a 10 milligram increase, half a millimole, within one generation. 1993 mother, 2015 daughter. Half a millimole increase in fasting glucose. And I'll show you 2-hour glucose during glucose tolerance test was 1.5 millimole higher. So this is how the comparison looks for first 150 deliveries. F0 mothers and F1 mothers. The BMI has increased by about 1 unit for these first 150 girls. Education has improved. Height, 5 centimeter gain in height within one generation. We saw between 1830 and 1980, Indian male had gained 0. Between 1993, parent and their daughter, 5 centimeter increase in one generation. BMI, 1 unit increase. GDM prevalence, massive increase if we use criteria, which of course I don't believe. Fasting glucose, 10 milligrams higher, 2-hour glucose, 31 milligrams higher. Insulin concentrations high, disposition index low. So these girls who were born small to a small mother with the rapid development are developing large gestational diabetes because they have low disposition index, which I showed you is present from 6 years of age when we first measured it. So I'm sure it was there before. It's a problem of intrauterine beta cell programming. And look at the babies. So this is now F1 generation mothers birth characteristics, which we have measured in our study, and this is the F2 generation female. There is a 250 gram increase in birth weight in one generation, unheard of in nutritional trials in pregnancy. This baby is heavier, is longer, head is bigger, abdominal circumference is bigger. But very interestingly, its fat measurements are not higher. Now, what I haven't told you is that we started a multi-arm V12 trial when these girls were 15-year-old. So 2 out of 3 girls are receiving B12. I am blinded to the trial sort of groups, that's why I can't tell you, but it seems it might help us to come out of the thin fat phenotype. I can't say this just now. As a scientist, I have to wait for the trial to be over. We have followed children born in my unit in the diabetic pregnancies. First 200, average age 15 years, children of diabetic mothers, 5% were already diabetic. 37% are pre-diabetic, only 58% are normal. And mothers who were not diagnosed diabetic in pregnancy, 20% of her children are also pre-diabetic. But there is clear doubling of the whole thing. Even children below 10 years of age, where we did not do a glucose tolerance test but a capillary test, their glucoses were higher. So this intergenerational transmission of glucose is obvious from very early life. And as I told you, it's more likely to be intrauterine epigenetic programming rather than genetics, though we are studying genetics of it. Is there a mix of the undernutrition and overnutrition? I'm going to show you for only one thing. This is a classic study from Mysore. Krishnaveni has developed the best cohort there. It's being followed up now into 20th year. The B12 deficient mothers in pregnancy had a higher BMI, they had higher prevalence of GDM, and they had a higher prevalence of diabetes 5 years later. So a combination of micronutrient deficiency and macronutrient excess is what their babies will face. And these babies are being followed up. So I developed this idea of dual teratogenesis, published it almost 10 years ago, that there is an undernutrition driven programming cycle which we see in villages, which is what I have described extensively, where we produce small thin fat babies. If they remain undernourished as adults, they remain insulin resistant, unlikely to develop full-blown diabetes. They migrate to the cities or are overnourished, then they get into the Norbert-Frankel or the Pima Indian type cycle, where there is obesity, there is gestational hypoglycemia, and then there is a double whammy. And that's how the association of birth weight with future risk of diabetes is U-shaped. Both small and big babies are at a higher risk. And these are the two pictures. This was the nutrient mediated, which they underwent, but they are now driving a fuel mediated, like I showed you in the last slide. My PhD students are really great. Someone took the challenge of setting up a rat model to replicate what I found in humans. So we now have a 50 generation undernutrition rat model, which we call Thrifty Jerry. And after 50 generations, we allowed them to eat normal, not over, for 2 generations. And they became grossly obese, and being rats and animals, we are able to look at epigenetic markers in all the organs. This is a cell metabolism paper talking about epigenetic markers in beta cells. And they did not erase in 2 generations. What was acquired by undernutrition for many generations did not erase in 2 generations. So that's the thank you slide to show you that we have a group called SNEHA, that's David Barker in 1990s. SNEHA is the oldest DOHAD society in the world. We are older than the International DOHAD Society. This is my hospital and department. Some of my staff members, that's my administrator. She also administers my life. She is my wife. So she is the only one who complained that we had missed 5% follow-up rate. 95% follow-up, she said, was not good enough. And these are the participants in adult age. These are my village staff. They have made this 95% follow-up possible because they are all residents in the villages. We have been funded by various agencies, predominantly by the Wellcome Trust, MRC UK. NIH has given us 3 grants and a whole lot of Indian agencies. So with that, I'm going to say thank you. And the idea is that there is a modifiable susceptibility to diabetes which develops intergenerationally based on exposures which cause a growth retardation or a growth expansion. So abnormalities of growth and development in utero due to environmental factors are strong precursors to future risk of diabetes. And rather than treating 55-year-old 34 kg per meter square IGT people with eat less and walk more sort of idea, we might think a bit differently. And the village women, if they had eaten more and walked less, would probably produce less diabetes. Thank you.